Hello everybody, I'm Lesley Jones and I work at Cardiff University, and I'm going to talk to you about DNA repair in Huntington's disease. I'd like to thank the organisers very much for inviting me to tell you about some of the research that we've been doing. So that was a picture of Wales, which is about 150 miles west of London. It's really quite pretty. But one of the things that I wanted to say about Huntington's disease, and I'm not going to go into the background because I'm sure you all know a huge amount about it, is that if you express the expanded repeat that causes the disease in cells or organisms, in models of most sorts, then pretty well everything you look at in those models will be different from the way it would be if you had not expressed that expanded repeat. And that presents a problem because then you don't know which of those downstream things to put right in order to treat the disease. So we took a very different view on this, and as part of the Genetic Modifiers of HD consortium, we looked at the effect of age of onset, because we know that CAG repeat length correlates really well with the age of onset of disease and accounts for probably about 60% of the variance in age of onset. And this slide illustrates that in a European registry population. And you can see that if you have 40 repeats on average, you'll get the disease at around 50 years of age. And if you have 50 repeats, you'll get the disease on average at about 30 years of age. And that's a big difference. But what you can also see on this slide is that at each repeat length, there's a huge range of ages of onset. And we know from the work of Nancy Wexler and in the Venezuelan consortium that actually about half of this is inherited. So it's genetically encoded. So we decided to take a rather different approach and look for what those genes that affect age of onset might be, with the view that if you can detect those genes and variants, you might be able to find targets, biological targets, that you can attack that will allow you to move people from being early onset to late onset, so you can delay the onset of disease. So that's why we did this, and it'll tell you this is in a study in people, and it'll tell you what's important in people. So we did a fairly standard type of genome-wide association. And what we did was we looked at the age at which people were predicted to get HD from their CAG repeat length, which had been measured, compared with the age at which they actually got CAG. And we used this difference, the residual, to do an association study to see if we could find any variants right the way across the genome in millions of variants that were associated with high or low age of onset after accounting for CAG repeat length. And this has been published, so I'm not going to say an enormous amount about it. But what we saw, and this is our most recent GWAS data, and this was published in 2019, what we saw is a number of hits in the genome, which is what you want to see when you do a genome-wide association study. And the hits that I've highlighted here that rise above the genome-wide significant line, the red dotted line, are all to do with DNA repair. And we'd had an intimation of this from our earlier genome-wide association study. Most of these genes, PMS1, MLH1, MSH3, PMS2, and LIG1, are known to be associated with mismatch repair of DNA. But FAN1 is also a DNA repair gene. However, it's classically thought to deal with interstrand crosslinks, which are hugely deleterious to cell replication. However, we also know that it interacts with MLH1, and I'm going to come back to that later in my talk. There are a number of other genes that we found whose significance we don't understand nearly so well, and I'm just going to come back to these briefly towards the end of my talk. We also found a hit in Huntington's, and we've taken out the effect of the CAG repeat in these studies. And we now know that this is due to the fact that we have different types of flanking sequence in the Huntington's gene. And I'm going to talk a little bit about that later in the talk as well. The other thing that we noticed pretty early on was that this was not just an effect confined to Huntington's disease, but that we also saw it in a number of other diseases caused by the same genetic mechanism, CAG repeat expansion, in a series of spinocerebellar ataxias. This was actually a very small study, and we looked at 22 SNPs in around 1,500 people. But nevertheless, we did see the same signal. And this is interesting because it says that it's the underlying repeat in the DNA that's important in whatever's modifying the age of onset of disease through this mechanism. We also did some studies that tried to use other ways of measuring clinical onset rather than, or measuring the clinical syndrome rather than age onset. So we did a study with Sarah Tabrizi at UCL, looking at genetic variants using a progression measure of disease or a severity measure of disease. And we thought that this might be more accurate than age onset. And this actually gave us a slightly different result because it gave us a really big hit in MSH3. And this has proven to be very interesting. It goes in, it's actually a gene of the mismatch repair pathways. And this was the first intimation that it was actually genetically associated with a phenotype of Huntington's disease. So what this says is that we need to focus back on the CAG repeats in order to understand what's going on here. DNA repair has been shown previously, actually quite a long time ago, to be important in Huntington's disease pathogenesis. And Messer noticed in 1999 that if you delete MSH2, a mismatch repair gene, from Huntington's disease mice, you prevent the somatic instability that had been seen in cells in Huntington's disease models previously. And that's interesting because we know that that instability, Vanessa Wheeler showed that that instability correlates very well with the age of onset of disease. So in the top two panels here, you can see both of these people have 47 repeats, but the person on the left has much more somatic instability of their repeat in brain, and they have an age of onset of 25, whereas the other person has an age of onset of 41 and much less in the way of somatic expansion of repeats. So we think that somatic expansion is likely to be really important to the disease, and hence DNA repair causing it is likely to be really important in the disease. So how does it work? Well, the answer is that we don't really understand the detailed mechanisms, but what we do know is that the CAG repeats and other repeats in the genome tend not to adopt the more standard beta DNA conformation. And so they adopt a number of different conformations in the DNA. And some of these are almost certainly perceived by the repair systems to be lesions in the DNA. So the machinery that deals with lesions comes along and tries to repair those lesions. And in that process, it cuts out some of the DNA and resynthesizes it. And that allows both expansions and contractions probably. But once you get beyond a certain length of repeat, you tend to favor expansions rather than contractions. So this means that it's very likely that what we're looking at here is a two-step mechanism of pathogenesis. So it's likely that in people with, in their germline with less than 36 repeats, you're not going to see expansion. But once you've got 36 or more repeats, then that, if you like, gives the repeat the license to expand. And it expands then into the pathogenic range in individual cells. And that length, which causes pathogenesis in individual cells, is likely to be different in different cellular populations, and this remains to be determined. But it's clear that the most vulnerable cells are the medium spiny neurons in the striatum, the caudate nucleus and the putamen in people, and it's probable that those medium spiny neurons have a particular vulnerability, and once the repeat has got beyond a certain length, it's going to cause disease. In a shorthand sort of way, I've written pathogenic protein here, but actually it's, although there is evidence that the expanded polyglutamine translated from the CAG repeats is toxic to cells, there's also evidence that there may be other toxic mechanisms at other levels going on here, which it shouldn't be ignored. So we have a number of genes highlighted in red on the right here that we found to have some genetic association with Huntington's disease, age of onset, or other phenotypes. And they belong to this mismatch repair machinery. And what happens is that the MUT-S dimers, including MSH3, detect lesions in the DNA. So the MUT-S alpha dimer detects usually single base changes, and the MUT-S beta dimer detects slightly larger changes, up to about 15 nucleotides. This, once they've detected that lesion, or so-called lesion, they attract the MUT-L dimers, and those MUT-L dimers attach to the MUT-S dimers and cleave the DNA. The cleaved section is then removed, possibly by XO1, which we find significant in some circumstances, and it's resynthesized by a polymerase. We don't know what the identity of that polymerase is, and ligated probably by Lig1. So you can see that we've got a lot of implications for the DNA repair machinery being important in altering the age of onset and progression of Huntington's disease here. So that's fine, but how do we convert these genetic findings to biological mechanisms and find targets for novel therapies? Well, the idea is that we're going to try and move people up the trajectory from having earlier onset to having later onset by using these as targets. And there are a number of ways we can do this. First of all, we need to ask, are the signals due to gene expression? So is it the gene being, the amount of the gene product being titrated up and down? And we can do this using various imputation methods called transcriptome-wide association studies, which we've tried. And the other advantage of this is it also gives you a direction of effect. So this work was done by Peter Holmans, and what you can see here, if you concentrate on the top two lines of the spreadsheet here, is that FAN1 has a very strong signal, and so does MSH3, and they act in opposite directions. So for FAN1, increased expression correlates with later onset, and for MSH3, increased expression correlates with earlier onset. Some of the other genes are much less clear-cut. And the other interesting thing is if you look at this in various expression databases derived from cortical tissue, large databases, you can see that about half of the FAN1 heritability, most of the common but not the rare signal, is due to changes in expression. Whereas for MSH3, most of the heritability appears to be accounted for by expression. And that means that there's probably other genetic variants in FAN1 to find, but there may not be much in the way of genetic variants in MSH3 to find. We know that MSH3 knockout prevents somatic expansion in mice, and we also know a little bit about the mechanism, because if you knock out the ATPase domain, Bob LaHoo showed this in his system. If you knock out the MSH3 ATPase domain, then you actually prevent or substantially reduce expansion. We also know that MLH1 knockdown reduces somatic expansion. In fact, once it's knocked out, it completely ablates it. And we interestingly also know that it's involved in the expansion of other repeats, including the CCG repeats in Fragile X and the GAA repeats in Friedreich's ataxia. And they actually are much longer repeats. FAN1 interacts with MLH1 and protects against repeat expansion, and we can see this here in Karen Usdin's very nice Fragile X mouse model, where you can see that if you knock out FAN1, you get a larger expansion of the repeats in the hedgehog plots down on the bottom right here. So that's interesting because it implies that somatic expansion is likely to be going on pretty well from the early stages of the disease. The implication is that it will be going on at different rates in different cells right the way through development. And that means that it's likely to be a mechanism that can be tackled before very much damage occurs, although that, of course, has the same issues as the Huntington lowering trials encounter, that, you can't do the trials in people who don't currently have disease, but people are working on trying to overcome this. But we were interested in whether there were any more large genetic effects, so genetic effects of large effect size, and we chose to exome sequence the age of onset extremes in our cohort. So we sequenced about 500 people, with the idea that any protein coding variants were likely to be of larger effects and thus more detectable. And these data have been published on bioarchives. The first thing we noticed, and the thing that was responsible for the hit in Huntington, was that if you have two CAA-CAG, repeated CAA-CAG after the pure CAG track, that you actually see a very marked increase in the ages of onset, plus 18 years. Whereas if we don't have any CAA codons after the pure CAG tract and go straight into the CCG codon, then actually we see about 18 years earlier onset. So there's two things to comment on about that. One is that not all of these effects were detected in the GWAS. They exist on other haplotypes. So to find them all, you need to sequence the repeat region in detail. And the second thing is that part of this effect is due to the miscalculation in the repeat length, because most people do a PCR, which does not detect whether you've got intervening CAA codons. But not all of it is. So there is still a residual effect of the structure after flanking the CAG repeat. It's not just a an effect of miscalculation. There's something else going on here. And that implies that the structure of that CAG, pure CAG repeat, might be different. So there's a number of genes involved in DNA damage and repair, but I'm only going to talk to you about FAN1 and MSH3 here. So this is a cartoon of FAN1. And in FAN1, we see 28 damaging variants and 1 loss of function variants in people with early onset, whereas only 11 damaging variants in people with late onset compared with that predicted by their CAG repeat length. And what you can see here is there is some segregation across the protein. So people with down-pointing red arrows have early onset, and with down-pointing green arrows have late onset. And you can see there are two collections of early onset arrows, one in the DNA binding domain of FAN1 and one in the nuclease domain of FAN1. For MSH3, we don't see very much in segregation in the damaging variants, but we only see loss of function variants in those with late onset. This is not significant, but we had quite a small sample. So these are the variants that we saw, and we had some of them made as recombinant proteins to do some biochemistry to see if we saw effects on the nuclease domain. And in fact, when we do nuclease assays with these, so FAN1 preferentially cuts a 5' flap, so we can see when it cuts by looking for the product, the labeled product here. So here you can see the product, and you can see that with the wild type, you see a product here. And then if you use some of the late onset variants, you see much less product. And in particular, the R507H variant, which is the top hit in our GWAS, causes what looks like a complete ablation of the nuclease activity. That's quantified here. And what you can also see is that the level of nuclease activity correlates with the age of onset of the subjects. And in order to find out what was causing this, we looked at some work in iPS cells done by Jasmine, and she looked at cells that originally had a 109 CAG repeat. She knocked out FAN1, and she saw that when you knock out FAN1, you get more expanded repeats. But also when you inactivate the nuclease domain, you see very similar amounts of expanded repeats. And that's shown here. And what you can see here is that this is FAN1 knockout in yellow, and then FAN1 D960A homozygotes in blue. But if you have D960A heterozygotes, they look as if they're in between the two. These are iPSC cells on the left and neuronal type cells on the right. And what you can see here is that we see the same effect in both dividing and non-dividing cells, although the expansion is slower in the non-dividing neuronal cells. This is interesting because there's been 2 papers very recently published showing that FAN1, the interaction of FAN1 with MLH1 is critical to the promotion of expansion, but that FAN1 nuclease activity is also critical to the expansion of the CAG repeats. So we're homing in on the mechanism here. We've also done some work in animals, which I'm not going to talk about in very much detail. But here you can see that essentially the tail length in all of our FAN1 animals, including the FAN1 knockout homozygotes, the CAG length is the same, and that's shown in the sort of grey under here. But with FAN1 knockout, you can see you get this very long tail of peaks to the right, and you get a much bigger expansion index. And when we look at these, you can see that they are significant. So non-CAG control, we had the CAG repeats were different starting lengths, so we took some of the really outlying lengths out and redid the analysis, but they're both significant. Non-significant in the tail and significant in the striatum here. So it's the striatum on the right and the tail on the left. And the final thing I'm just going to mention is some work we've done on TSOG1, which is also on chromosome 5. And what Sergey in the lab has found is that there's a repeat section in TSOG1 that's coding that is tagged by this SNP. And we find that the repeat is actually the thing that's causing the that's best associated with the age of onset of disease. This is significant. This is also on bioarchives, and we find that we get 1 year earlier onset for each additional hexamer in that repeat. So the canonical repeat is 6, and the commonest repeat, the alternative repeat, is 3 hexamers, and that is tagged by the SNP. We don't know what TSOG1 does, and it could operate through somatic expansion because it's known to operate in transcription. It appears to have a role in splicing, and it may be that it stabilizes RNA DNA hybrids during splicing and transcription, and this may predispose the DNA to being more exposed to being cut. So we think that we've definitely seen things that act on the CAG repeat, and possibly we've seen things that are to do with transcription and RNA processing that may play into the exon 1 mRNA production that Jill Bates has seen and published about. So DNA repair pathways are interesting. This is taken from Jessica Brown's review, and it's a series of pathways, double-stranded break repair pathways and single-stranded break repair pathways. And these have been used in addressing cancers. This is a cancer paper, cancer review. And those that are coloured in red here have programs looking at producing inhibitors for them. So there's already some chemistry in this space that we may be able to leverage in Huntington's disease. Because we think there's likely to be a vicious cycle going on here. We think that you get DNA damage and repair, and this causes the repeat, probably very rarely, to expand, because you're likely to see that the damage is repaired efficiently. However, as the repeat gets longer, it itself causes DNA damage, and we know this from previous publications and experimentation. And that DNA damage is likely to induce repair activities, and that repair activity is likely to induce more expansion. So you end up in a toxic cycle where you get increased DNA damage, which leads to increased somatic expansion. This may be cell specific, which is maybe why we see the effects in particular cells. So I'd like to thank the patients, families, clinicians and researchers who made these studies possible, and all of our funders. We're really grateful for this support, without which we couldn't do the work. And I would very much like to thank my group and my collaborators who also took part in this work. And thank you very much for listening. Hi. So I thought someone was going to play a cartoon now. I don't know whether the audience can hear me. Somebody just told us that you can hear me. Okay, so I've got some questions here. Chris Ross asks, do you see greater somatic expansion in the CAG 109 line compared with others? And if so, do you know why? I assume that Chris means that, do we see expansion in other lines? And the answer is that we actually tested quite a lot of different cell lines, including non-stem cell lines, looking for a line in which we could see expansion so that we could use it to generate an assay for expansion. And this was the only line so far in which we've seen substantial expansion, which we can characterize reasonably well, so that it gives relatively consistent results. And that's why we use this line. We are looking at others. Certainly in the HD stem cell consortium line that was around 60 repeats, we saw no expansion. And that may be to do with the length of the repeat, because longer expansions are more inclined to expand. And Chris also asks, when does the somatic expansion occur, during development or adulthood or both? And the answer is that I don't know, but I'm currently doing an experiment in mice, which we hope might provide the answer. So we are going to look at, everything from embryonic cells onwards, and we'll keep you posted. Martha Nance asks, does this model suggest a person with 25 repeats might occasionally undergo the somatic expansion that would lead to HD? Is there a reason to think that 36 is a magic number? So, Martha, I don't necessarily think that 36 is a magic number, and that may account for why there is that grey area between 36 and 39, where you don't necessarily get complete penetrance. And I think it's possible that there may be occasional people who, for instance, might have a very deleterious FAN1 variant who may show expansion, but we don't know. Rajiv Kumar asks, is there an increased risk of cancers if you reduce the activity of MSH3? And the answer to that is that we think probably not, because actually the evidence for MSH3 loss causing cancer is quite is not very strong, but clearly it is something that needs to be taken into account. But I think if there's still some, I think that there is, still some. Oh, I've got myself lost now. Sorry about that. We think that there is still likely to be some MSH3 activity. Going on. I think we would aim to just reduce it rather than ablate it completely. The precise interaction of FAN1 and MLH1, I think you should read the two papers from Sarah Tabrizi and Alex Sartori's lab, because they give a really the most up-to-date explanation of how that interaction works. And it looks like the effect of MLH1 is dominant to that of FAN1. Is the CAG repeat expansion different in the striatum and cerebellum? Well, this is not our work, but yes, the answer is that it does appear to be much less in the cerebellum. Does the CAG expansion does occur in other tissues? You see it in liver, for instance. What's the latest thinking on whether HD gene carriers will get additional tests for modifier genes? Well, I think that we don't, the genetics is not firm enough for us to do this yet. It's possible that it may be, they could be added and give a more accurate age at onset, but I think it's more likely that they are likely to be used to refine, you know, things like clinical trials initially. So I have to say that I would be reluctant to introduce them now. I think we need to know more. And it is true that some of the coding variants that we found in FAN1 do have a very big effect. But we sometimes, you know, they can be private, what we call private mutations. You only see them in one person, and that's really hard to predict. So I think that you could have a panel and generate some sort of improvement, but it still would not be completely accurate. So I would be quite wary about doing this at the moment. And I think I've now answered all those questions. Okay. Oh, what do I think the mechanism of cell death caused by somatic expansion? So I don't know what the mechanism of cell death that's caused by somatic expansion is. And it could be the toxic protein that is certainly can be toxic to cells once the polyglutamine tract expressed is beyond a certain length. But actually, I'm fairly agnostic as to what the mechanism of cell death is, because I think it's likely to be a bit different in different cellular populations, and I think that's still to be worked out, because people suggest, you know, RAND translation and RNA differences in the RNA, which certainly might occur. So I think that it's not the somatic expansion per se that causes the cells to die. It's the RNA and protein downstream of that expansion that causes the cells to die. Do you think? Yes, I do think that the cells that I'm asked, you think that the cell selective somatic expansion is related to FAN1 and or MSH3 levels? Yes, I do, but I think it's probably related to other things as well. I think there's probably a polygenic effect here, and it's possible that it isn't simply genetics, you know, because other things cause DNA damage. So it may be, it could be exacerbated by other things that might damage the DNA. It's a possibility, but I don't know. And I think that is now the last question that I have on here. Unless anybody wants to ask anything else. So thank you very much, everybody, and thank you for your interest. And let's hope we have a treatment soon.